All right, Assalamu alaikum everyone. Welcome to this PAL session. I hope you're doing well. My name is Maryam Altayeb. I'm a medical intern, and some of you guys may know me from pre-med, and some of you may know me from the talk we gave you guys in the beginning of the year for this little booklet that we worked really hard on. Let's Speak Made Easy. If you didn't get a copy today, there are some backstage in PHA. But I'm going to start sharing screen. First of all, I just want to start off. It's very intimidating. It can tend to be very tough, but I promise you we're gonna get through it. Also, I know this particular lecture has a lot of slides, but don't worry, a lot of them are pictures and hopefully, we're gonna go through basic concepts together. And if you have any questions, please just write them to me in the chat. I'm happy to stop and repeat any concept that's confusing. So I know this might feel very daunting, intimidating, especially MOL. There's a lot of terms, everything is new, but hopefully we can make it very easily digestible for you guys. So Bismillah. Today we're going to be talking about DNA to RNA transcription. Okay, so these are our general objectives that we're going to be going through, or the flow of the lecture. First, I wanted to start off with a quote, because this is something I wish I had emphasized to myself in first year. Which is when we give ourselves permission to fail, we at the same time give ourselves permission to excel. I know everything is new, everything is scary, but keep in mind that failure is a part of the process. If you don't do as well as you wanted to, you can always do better. You can always learn from your mistakes, and that's something very important. We tend, especially as med students, to be very hard on ourselves and think that mistakes are the end all be all of our entire career. But on the contrary, mistakes are actually much more beneficial than you think they are. So please don't be afraid to get stuff wrong, ask questions. You're here to learn at the end of the day. So I just want to make sure you guys benefit from that. So first, let's talk about the central dogma of molecular biology. So what does this mean? Well, a lot of the processes in your body, if not all processes in your body, happen because of proteins. So how do we get from our genetic material all the way to proteins? That's what the central dogma of molecular biology tells us. So we have our DNA, which is the genetic material found in our cells, and this is our genetic blueprint, okay? And so in order for our cells, for us to keep living, our cells need to divide, right? So DNA is going to copy itself, and that's called DNA replication. But DNA can also be changed into RNA, okay? And that process is called transcription. And that's what we're going to be focusing on today. And why do we need transcription? Because it basically allows this DNA to be turned into RNA, like a blueprint almost. And then this RNA is what you follow to make your proteins. So it's kind of 3 steps. So you have your DNA to transcription, and then transcription takes RNA to protein, and that's called translation. And so this is how basically all of our cellular processes work. And this is just visually what that looks like. This is our DNA here. And then by transcription, it's turned into RNA, which you'll notice is a single-stranded molecule. We'll talk about the differences in a second. All right. And then through transcription, that happens. And then through translation, that RNA is turned into protein by codons. A codon is basically a term that you might come across. Let me just put the laser pointer. Yeah. So a codon is just when you have 1, 2, 3 of these base pairs grouped together, and that turns into one protein at a time. Okay, and so we have different kinds of proteins that I'll show you guys in a second, but just remember there's adenine, thymine, cytosine, guanine, and uracil. And you'll find that some of my slides are dark in color and some of them are light in color. These are just the exact slides from your molecular medicine course. I want to make sure to include them so you guys can follow along. What I'm basically going to do throughout this lecture is explain on my slides with the pictures, and then we're going to go through these together a lot more briefly, just to show you that everything is already explained and you already know what's going on. So a codon is a sequence that consists of 3 nucleotides. We just said that, okay? mRNA is the molecule that presents codons to ribosomes. We're going to talk about mRNA in a second. DNA serves as a codon storage molecule. How does that make sense? Well, this DNA is going to be turned into RNA, right? So it's almost like our DNA is storing all of these codons, all of this genetic information that is turned into RNA. Guys, can you please make sure to mute when you join? Thank you. All right. And so ribosomes translate codons in sequence into a chain of amino acids. That's what we said, right? So you get your DNA, that goes to RNA, that RNA goes to proteins, and each three base sequences or each codon turns into a specific protein. Okay. And once translated, proteins are also often post-translationally modified. It just means you need to fix them up a little bit. And proteins consist of one or more polypeptides. Okay. So basically, what they're saying is everything we just described, all right? And that proteins are just a chain of these different molecules, these different amino acids that form a polypeptide. This is just basics, okay? So now, what's the difference between DNA and RNA? So the D in DNA is basically for double-stranded, okay? And it's deoxyribose nucleic acid. So I just want you to remember that it starts with a D. And so this would be down here, our DNA molecule, for example. So it's double-stranded. It has a deoxyribose sugar. I'll show you what that means in a second. And it has thymine instead of uracil. RNA is ribonucleic acid. So the R for ribonucleic acid means there's a ribose sugar, okay? And it's a single-stranded molecule. We just saw that here, right? So DNA is double-stranded. It's in a double helix pattern, and RNA is single-stranded. But here, you're going to have your thymine, and here you're going to have your uracil, which would be, here in orange and here in red. All right. So this is just another visual representation of that. So we said DNA is double-stranded, deoxyribonucleic acid, and the base pairs that are here would be thymine, okay? And thymine will usually pair with adenine. However, in our RNA, anywhere we would have an adenine, instead of putting a thymine, we would put a uracil. Okay, so that's just important to remember. Here you have U, you don't have T. Okay, and our RNA is a single-stranded molecule, and it has a ribose sugar. So this is our ribose, and this is our deoxyribose. You can notice the difference here, how there's no oxygen at this, here in this carbon. Here you have an H instead of an OH. Here you have OH and then another OH. All right, so I think it's a deoxygenated ribose. It has one less oxygen. All right, and so this is just, again, what it looks like from your slides. So our RNA structure consists of our sugar. Remember, it's called ribonucleic acid. So ribo is for ribose, the ribose sugar that we just looked at. Okay, it has a phosphate group and a nitrogen-containing base. Why? Because it's a nucleic acid. So what are the potential nitrogen-containing bases we could have? We said in RNA, we can have adenine, guanine, cytosine, or uracil. And what we just said is in RNA, uracil binds to adenine, right? But in DNA, thymine binds to adenine instead. So that's just important for you guys to keep in mind. They can especially ask different MCQ questions about this. And I have a few examples with a lot of MCQs throughout this lecture, so you guys can test yourselves, and hopefully it'll help clarify any misconceptions you have. So again, this is just what's in your slides, repeating it briefly. So RNA is single-stranded, and RNA is short. Why? Because it's coding for one gene at a time. DNA is all of your genetic material attached together. We know RNA contains ribose, DNA contains deoxyribose, and RNA can store information like DNA and catalyze chemical reactions. So, for example, you have proteins like ribozyme that can help assist in specific chemical reactions that are going on in the body. All right. And all right, so this is basically the important thing I want you guys to know for now. Remember, in DNA, adenine pairs with thymine, but in RNA, adenine pairs with uracil. But no matter what you go to, cytosine will always pair with guanine. So this is just an example of this. So this is what it would look like in DNA or RNA. You guys tell me. If this has thymine in it, would this molecule hypothetically be a DNA or an RNA?
DNA.
DNA, exactly. Thank you, Abdurrahman. So if you ever see a T, then you know it's a DNA molecule. If you ever see a U, then you know it's an RNA molecule. And so these are just two visual representations of how these would pair together. I just used to memorize it as ATCG in DNA and AUCG in RNA. There's not really like a rhyme or reason to it. You just kind of get used to it the more and more you see it. All right. So now we're going to talk about different types of RNA. But before we start that, do you guys have any questions? I know it was just revision of the last lecture, but if you have anything you want me to re-explain, let me know.
No, until now, everything is clear.
Okay, that's perfect. So now we're going to talk about different kinds of DNA. So in the beginning of the lecture, we mentioned something called mRNA. So mRNA is messenger RNA. So think of it as it's sending a message. It's your DNA being translated to send a message. And then that message is going to your ribosome to form your proteins in translation. So this is just one of the very important types of RNA you're going to encounter. You have small nuclear RNA, which process mRNA into its mature form in eukaryotes. We're going to talk about these in a second. As we go throughout the lecture. You have ribosomal RNA, which from its name makes sense that it goes towards ribosomes, right? So the ribosomal RNA combines with proteins to form ribosomes, the site of protein synthesis. So as you can see, all of these have different important bits and pieces for you to just get your protein at the end of the day. You also have transfer RNA. So from its name, transfer RNA or tRNA transports, transport, it's moving something from one place to another. So it transports amino acids to the site of protein synthesis. So all of these different kinds of RNA eventually play important roles in you getting your end protein. Okay, so this is just, again, what is written in your slides. Nothing scary. It's what we just talked about. Messenger RNA is sending a message, right? So it's transferring the DNA code to ribosomes for translation. And in the next lecture, you guys are going to learn more about this. You have transfer RNA. Remember, it's moving something from one place to another. So it brings the amino acids to the ribosomes for the protein synthesis to actually happen. Ribosomal RNA is making up your ribosomes. You have something called heterogeneous nuclear RNA. And we're going to talk a bit more about this later. I don't want to explain it and confuse you now. But it's basically the in-between steps when you're processing your mRNA. You have small nuclear RNA, which is, again, only found in the nucleus, and it participates in splicing. We're going to look into this a bit later. You have microRNA. So remember, if you see micro in front of anything, it means something really tiny, like microscopic. So they're short, non-coding, and they control the expression or repression of some genes during development. So we're going to talk, again, a bit more about this later. I just want to make sure you guys get the terminology across from them. And ribozymes, so ribosomal enzymes, are RNA molecules with enzymatic activity. All right. So just these words you're going to see thrown around a lot in these lectures. So just if you have any confusion, refer back to the slide, or if it's a bit easier, refer back to this table. All right. So that's for the first two parts. Now let's talk about gene transcription, which is going to be a very big bulk of this lecture. So now we know we have our DNA. Okay. Our DNA is an anti-parallel molecule. What does that mean? Anti-parallel means they're bound to one another, but they're going in the opposite direction. So if you have an arrow going up and then an arrow going down, these two are anti-parallel. Think they're going in opposite directions. And so when we say they're going in opposite directions, what does that mean? You have a 5' end of this lighter purple one, and then it's going to continue. Remember, it's anti-parallel, and then it ends in our 3' end. Okay, and then you have the opposite for the other strand. Instead of going 5' to 3', you're going 3' to 5'. And what do we mean when we're talking about 5', 3'? You're going to see that thrown around a lot. It's basically where the phosphate group lies in terms of this molecule. And the reason that's important is because when you have the phosphate group at the end, it allows some of the enzymes we're going to talk about to work properly. Some of them can't work in the opposite direction. It needs to have this molecule exposed so that it can actually go along the DNA or the RNA. And we'll see what that means in a second, okay? So what I want you to remember is the 5' to 3' strand of DNA, which you can see here, is the coding or sense strand. And we'll see why in a second. but this strand is our master blueprint, okay? Because it ends up looking like our new RNA molecule, right? But our other strand that goes the opposite way, 3' to 5', is our template strand. Sorry. I'm done. Okay, I think I'm done. This is what happens when you have cats in the house. Thank you. Okay, so let's go back because this is a very important concept for you guys to get. When I say something is a template, okay, it's something that I'm copying, right? So our template strand is the strand that we are copying, okay? But it is our antisense strand. So if I'm copying this template that's going this way, it's going to end up looking like this other strand, right? Because they match. They're anti-parallel and they're complementary. So our antisense strand is the template that we're using. But when we use this template, the new RNA molecule we end up making looks like the coding strand. And I'll show you again in the next slide. So if this is our coding strand, our coding strand is what our new mRNA molecule is going to end up looking like. And it runs 5' to 3' direction. So here, for example, A, T, C, 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 or whatever. And is this DNA or is this RNA?
DNA.
DNA, صح? Because we have our thymine here. So we know for sure no way we can have thymine in our RNA. So this is the coding strand of our DNA molecule, which you can see here. The one we talked about first. Okay? So what about our template strand? Our template strand is running the opposite way. Remember, they're anti-parallel. So T, T, A, G, G, etc. It matches the coding strand. But remember, the template strand is what we're actually going to copy off of. And our new part, our mRNA molecule that we're going to end up forming is going to look like the coding strand. Okay, it's going to end up looking like this. But instead of having a T, what are we going to have instead?
You're gonna have a U.
We're going to have a URL. Amazing. So now let's start to get to the actual process of transcription. So now we understand how these molecules go in different directions, what's coding, what's non-coding. And if you didn't, it's okay. I have more visuals that are going to help you a bit later. So the 3 stages of transcription are initiation, meaning you're starting the process, elongation, meaning you're making the molecule longer, and termination, meaning you're stopping this completely. And these are the exact same general steps as DNA replication. And they're just, what does non-coding mean? I'm going to repeat it for you again in a second, okay, Lama? When I get to the other picture, you'll see what I mean. So, no problem. So generally, when you're initiating something, you're just starting something. So this is going to tell us how to start our process of transcription. When you're elongating something, you're just making it longer. That's this here. So you're basically building our new mRNA molecule. And then termination, to terminate means to end something, right? Like if you're getting terminated at your job, you're getting fired. So you're ending your contract with those people. So here it's similar. You're stopping the process of transcription. Okay? So how do we know, or how does our body understand where to start and stop transcription? So you have specific sites in your DNA that are considered start sites. So you have specific start codons and stop codons. But what I want you guys to know, because you're going to encounter these terms a lot, is you're going to see things called upstream promoter, downstream terminator. What does that mean? Well, if this is the gene that we're looking at in this orange color, anything upstream is going to be before this gene starts. Okay, so the promoter is upstream. So when you're promoting something, you're saying, okay, this is good, keep going. And it's like you're encouraging it. So our promoter regions are actually guiding our enzyme called RNA polymerase that we'll talk about in a second, and telling RNA polymerase, hey, I want you to start here. This is where you should be. So it's promoting the start of transcription. And then you have your terminator. So your terminator is basically where your RNA knows, or your RNA polymerase knows, okay, خلاص, here is where I stop my transcription process. Okay, so the promoter region determines where your transcription of DNA begins and turns into RNA. Terminator tells you, okay, here's where you should stop. All right. And so in transcription, we have specific promoter regions that we just talked about right here, right? Remember the region that's like a green light telling you, hey, you should start here. So these promoter regions have specific sequences. They're just non-coding, meaning they're not coding for our transcription, right? They're not coding for RNA. And they're repetitive, meaning it's just the same 2 or 3 base pairs again and again and again. So here, for example, it's TA, or the Tata box, as they call it. So this promoter region, or our repetitive non-coding sequence, tells us, okay, here is where I need to start. Okay? You guys, give me 1 second. I'm just going to get a tissue. Sorry. Okay, I'm back. I'm sorry, my cats really got to me today. So when we're starting our transcription process, right, we're initiating transcription. How do we know where to start? Because of these promoter regions, they have repetitive non-coding sequences, for example, the Tata box. All right. So you begin your process by unpacking the DNA and dehelicization, which means you're just unwinding the double helix. You're exposing individual genes. And when your RNA polymerase enzyme sees this promoter region, it knows, okay, here's where I'm going to start the process. Okay, and so these are different types of promoters that you might come across. So you have the Tata box, which is what we just saw. You have something called the CAT box, GC box, CAP site. So these are just different sequences that will be repeated again and again and again. And it tells, again, the RNA polymerase that, hey, you should start here. So what it says in your slides is eukaryotic promoter lies upstream of the gene. But we already know what that means, right? Upstream just means before the gene, right? Downstream would be after the gene. And so there's a variety of different promoters that you can find in eukaryotes, so human cells. All right. And yeah, essentially. Yeah, go ahead. Do you have a question?
Does the promoter get transcribed?
No, it's a non-coding region. That's what it means when we say non-coding means we don't transcribe it.
Okay, thank you.
It's just like, imagine, like, in the airport when they wave like the flag and tell you, hey, you should come land here. Think of it as the same thing. Hey, you should start your transcription process here. Yeah, go ahead. You have another question?
Excuse me? Can I ask a question?
Someone is raising their hand. Yeah, sure, but just wait for there's someone raising their hand that has a question
Hi.
to ask.
I just have a question. Is there like what does upstream mean? I'm just confused.
Okay, I'll show you again. So, okay, you see where this is the gene in orange, right? Upstream means before the gene starts. So this promoter is considered upstream to the gene. It's just before it. It's just another way of saying before. Okay? And so when you say downstream, it's the opposite. It means after. So this area in red. Okay, does that make sense?
Yeah, it makes sense. Thank you.
You're welcome. Someone else had a question? Think on
Yes, here in this slide. What's unpacking DNA, please?
Okay, so your DNA is really, really tightly wound around each other, okay? Because we have a lot of DNA, and if it was unwound, it's basically way too long for us to be contained in the bodies that we are. So it's condensed in something called chromatin. So unpacking just means you're unwinding it so that you can access this part of your genetic code. Imagine you had like hair that was really, really tightly braided, and you had to fix a part, so you're unraveling it to get to that one part. That's just what unpacking means. So you're removing the really, really tight coils and unwinding them in order to get access to the parts of the DNA that you actually want to transcribe. Does that make
Okay,
sense, or do you want me to repeat it again?
It's the same as what was the word?
Dehelicization, yeah. So remember, our
With what
DNA
it
is in
is.
a double helix, the two strands that are wrapped around each other. So dehelicization means you're removing the double helix. Instead of having it tightly wound around each other, you're unwinding it, you're opening it so that you can actually use that part.
Oh, so
So
they are different.
unpacking is yani, okay. Imagine, okay, I'm gonna give a silly example. Indomie. Hello? When you get your Indomie out of the packet, it's already spiraled, صح? But they're really stuck together, right?
Yeah.
But unpacking would be when the Indomie starts to like fall apart. Yani they're not stuck together anymore. But لسه it has the spiral shape, صح?
Okay, yeah.
Dehelicization would be like لو مسكت حبة الإندومي وسحبتها خليتها like مستقيمة. You made it straight. Do you get what I mean?
Uh-huh. Yeah.
So your DNA is packed into chromatin, and chromatin is like when the Indomie is stuck together. All right. And then the helix is when the Indomie is كده wound around itself.
Okay,
فهمت
thank
علي?
you.
So unpacking is like you open that, and then dehelicization is when you remove the helix.
Thank you. Thank you.
I have a like, I know you're in the camera. Oh, ignore my sister, please. All right, so these are the different types of promoters that we have. So this is, again, what we just talked about in words. Hello? The RNA polymerase is the main enzyme, and it does all of the steps in transcription. So it's easy. There's only one enzyme you need to know, and it's called RNA polymerase. Make sense? It's going to help make our RNA. So it controls transcription. It searches for the DNA initiation site, اللي هي an upstream promoter that we talked about, unwinds a short stretch of double helical DNA to produce a single strand of DNA template. Okay. And then the RNA polymerase starts to build the RNA chain. So it's doing everything. It finds out where should I start. It starts to actually do the elongation process, or adding our different bases together. And it builds the RNA chain, and then it terminates. You'll see what I mean in a second. This is basically the initiation process. So remember, this is our condensed chromatin. We're going to unpack it. We're going to unwind it. And then the RNA polymerase is going to find the promoter region, for example, the Tata box. Okay. And it's going to start to actually make our different base pairs and put them together. Okay. So it's going to start to make our RNA molecule. And so when it binds and goes to these regions, it forms what we call a pre-initiation complex. And so that kind of is like a green light telling the RNA polymerase, okay, then let's start working here. So this is again from your slides. So we said RNA polymerase is the enzyme that controls transcription. It gets to your initiation site, it unwinds, it starts to add the different base pairs to make our RNA, and it detects our termination signals. So it's basically, this slide is telling you RNA polymerase is doing all of the work. All right. And so when we're talking about eukaryotic transcription, you have something called transcription factors. So transcription factors are proteins that bind to DNA near the start of a transcription gene. What does this mean? Wait, does the unwinding take place before the initiation site or after? At the initiation site, because you need to know where to initiate, and if it's tightly wound, you won't figure out where it is. So at the initiation site, it unwinds. No problem. So transcription factors are proteins that function in combination to recognize and regulate transcription of different genes. So the reason this is important is, or what we're basically trying to convey to you here, is that you have different factors that can affect whether translation and transcription are happening or not happening. And these are just different parts of your genes that are important for you to know about. For example, transcription of specific genes can be affected in diseases like cancer and diseases like autoimmune diseases. And so these are different proteins that can come and influence whether or not and where or not you start your transcription. So remember how I just told you in the slide before, you have this pre-initiation complex that tells our RNA polymerase, hey, you should start here. It kind of almost helps it find the initiation region. That's kind of what it's doing here. It helps you find the Tata box, binds to different regions, and almost helps anchor our RNA polymerase so that the whole process can start. So when you look at transcription factors, this is basically what they're talking about. They connect with our RNA polymerase so it can initiate transcription properly. Okay, it's like if we're going with the example of when you're at the airport and you see the people that are waving the flags to tell the pilot that he should park here, okay, or he should not park here, you know what I mean? Lower the plane here or start landing. Think of your transcription factors as the lights that also help guide it to its destination. Okay, so all of these are working together for us to actually start the process of transcription. And so once we start the process of transcription, we're going to go into elongation. So remember what I told you before, and if anyone was confused about it, I'm going to clarify it now. Our DNA runs in two directions, right? It's like a spiral around itself. You have your coding strand and you have your template strand. All right, just general terminology. When I say I'm using this as a template, I'm using this as a guide, I'm following this template, okay? But when we say our coding strand, that's basically what the RNA ends up looking like. You have something, or you have two strands, one that runs 5' to 3'. Remember, that's the direction of the phosphate, or 3' to 5'. And so when we have RNAP here, just means your RNA polymerase. Okay, so RNA polymerase can only work in the 5' to 3' direction. What does that mean? That means you guys can see this in blue here. The new RNA that we're forming has to be 5' to 3'. So when I'm forming my new RNA, okay, I'll show you guys. I know when is it 3' or I'll show you in a second, but just hang in there, okay? So if I tell you that my new molecule, my new RNA is gonna be 5' to 3'. And we know that it has to be copied off of something. So it has to be copied off of something that is working or that is running 3' to 5'. That is what our template strand is. يعني زي ما هنا, if this is running 3' to 5', okay, the opposite strand, it's anti-parallel. Remember, if one is going up, the other is going down. So if this is 3 to 5, that means this has to be the opposite, right? 5 to 3. And they're anti-parallel, they're going in the opposite direction. So the new molecule that you're forming has to be 5 to 3 because you're copying it off of this end. But when we say the template strand, this is the strand that I'm copying off of. Okay, this template strand is running 3' to 5'. The coding strand basically means it ends up looking like our new RNA. And you can see this is running 5 to 3. This coding strand at the top is also running 5 to 3. And if this, well, I'll show you in the next slide. Maybe this is easier for you guys to visualize. So here, our RNA polymerase is working on the template strand. It's copying our template, right, or our non-coding strand. And it's using the template to form something that looks exactly like this top strand, the coding strand. Okay, so you can see how it's basically the exact same. The only difference is in RNA, we're going to have a U, and in DNA, we're going to have a T. So when I say this is coding, it just means, you see how like these look exactly the same? This is blue, this is blue, this is green, this is green. Because they're being copied off of the same template. That's what they're trying to describe. Okay, so I'm going to repeat it again. In your DNA, it runs anti-parallel. يعني واحدة جاية فوق, واحدة جاية تحت. They're opposite to each other. Or maybe this is a better example. Okay, they'd be like this, opposite to each other. Okay. So our template strand, remember, a template, just as a general word, is what you're using to copy off of something. Hello? So our RNA polymerase molecule is going to copy the template strand to make our new mRNA molecule. And the mRNA is going to run 5' to 3'. They mean like who? Like our coding strand, which is basically its sister strand, what it ends up looking like. And the only difference is in our RNA, we have uracil instead of thymine. Okay, because thymine is only present in DNA. All right, I hope this makes sense for you guys. So template is what we're using to copy, and RNA polymerase can only work in 5' to 3'. Yani what it's copying off of has to be the opposite, 3' to 5'. Okay, and so this new mRNA that's formed ends up looking like this. And the reason we call it our coding strand is because this is essentially what's going to be used to code for our proteins. That's why it's considered coding. All right. I'm going to show you guys another example that's going to help you understand it better. And I want you guys to try and solve this with me so that we can make sure that you understand the concept. So we have our template strand, which runs 3 to 5. And we have our coding strand, which runs 5 to 3. So what would our new mRNA sequence be? We said when we're copying our mRNA, who are we going to end up following? We're following the template, right? So we can say, if this is a T, it's going to bind with A, right? So our new sequence is going to be what? AA. Here an A binds with a U in RNA. So AAU. And then G binds with C. So CC. T binds with A. So again, A, G, U, T. So all I'm trying to show you here is when you copy the template strand, that's what's giving us our new mRNA molecule. What's another way to do it? We know that our new mRNA sequence is basically the sister of our coding strand, right? But what would the only difference be? Instead of a T, what are we going to have instead? A U, right? So I can get you this MCQ, or you guys can get this MCQ in your exams in 2 different ways. If I tell you this is the sequence of the template strand, okay, what is our new mRNA sequence? All right. Then you can basically match it up. So a T here, you would have an A you would have a U, you would just start to make the new sequence. But if I told you this is the coding strand, what I would do is instead I would take the exact same sequence, but instead of the T, I would put a U in its place. Okay, so this is what it's going to end up looking like. Mubarak, you have a question?
I just wanted to note something. The doctor said in the lecture that he might give us a question in the exam, such as that he gave us a template strand and he doesn't tell us if it's 3 to 5 or 5 to 3. And he said if he doesn't say anything, it's going to be from 5 to 3. So we must flip it, then change it to mRNA to get the mRNA.
Okay, good to note. Usually if he says template strand, like if he specifies this is my template strand, then you'll know to pair it up, like you mentioned. يعني تقلبوها you pair it up. If he mentions to you that this is the coding strand, يعني coding strand is its sister, right? Like حتى if you notice هنا in the coding strand and the mRNA, they look almost exactly the same, but the only difference is the U. صح? So keep that in mind, because they can trick you. They can tell you this is my coding strand, what is my mRNA. يعني في الcoding strand ما راح تقلبها. فهمت علي? You're just going to take the same sequence. But keep in mind the terminology they get you guys in the questions. This is just an overview. Is that the codon length? This is just an example. يعني it's not the codon length is the length of the gene, but there's still more modifications that we're going to do later that you guys are going to see. But this is a very important concept, and I have a practice MCQ for you in a second. I'm just going to go through the slides so that you see in okay? So overview of transcription. We said, how do we know where to start? We have our promoter, and it's our non-coding regions like our cat box or our tata box or whatnot, right? And this is where our transcription factors bind, and then bind to RNA polymerase and say, okay, let's start the process of transcription here. Then you have the actual gene, and there's 2, right? You have your template strand, which is what we're following. and you have your coding strand, which is what it ends up looking like. Okay, and then you have your terminator sequence, which is where you cut everything off. This is everything we just talked about. Again, this is just how it's written in your slides. The template strand is called antisense. It runs 3 to 5, and now we understand why. Because RNA polymerase only is able to form our new RNA 5 to 3. And it has to copy something the opposite direction, 3 to 5, for it to actually work properly. And then you have your coding strand, which is the thing that looks like what you're transcribing newly. Well, basically this coding strand looks like your new mRNA. Template strand is what you're following. All right. So I want you guys to solve this MCQ. I'm going to give you a couple minutes. So again, keep in mind the terminology I'm using. The question is, an investigator is studying the transcription of DNA in a mouse model. Which of the following best describes the mRNA sequence if the DNA template strand reads 3' ATCG ATTCA 5'. So remember, I'm giving you guys the template strand. That's the important thing here. Hadi, take your time, take a couple minutes. Okay, so you guys are doing great. Your answer is correct. But before we say that, okay, the answer is E, let's understand why. We said the DNA template strand reads 3' ATC, etc., etc. And can anyone tell me what a template is, or what is a template strand? Yeah, go ahead, unmute. Go ahead, Mubarak.
It's the strand that we copy off of.
Exactly, it's the strand that we copy off of. Hello? So if we're copying off of this strand, what will we end up getting? Let's find what's the complementary base pair to A. Remember, we're talking about RNA. Okay, so it's instead of a T, it's going to be a U. You're going to have U, and then T is going to be A, G is going to be C is going to be G, etc., etc. And our new molecule is going to run from the 5' to 3' direction. You guys are right. There's a mistake in the typing here. There should be it should be AA. So I'll make sure to fix that. But you guys understand the general concept, صح? Because this is basically how they're going to get you your questions regarding this specific concept. So if I tell you this is my template, yeah, go ahead, Yasser.
فلو بيكون في template, يعني if they give us the template, should we just flip the entire thing? يعني
اوكي،
نسوي لها
خليني اكتبها معاكم. حلو. Okay, wait. Let's use the pen. So this is our template strand. يعني our template is what we're using to copy. So we're gonna find what matches it. What matches A?
U.
U. حلو. What matches T?
A.
A. And we'll basically keep going. What matches G?
C.
C. Ignore like I'm drawing with like the mouse, so it's gonna look really ugly. بس مش حالكم. A is gonna match with U again. U. And then هنا مفروض A, and then G,
U.
and then U again. What they mean when they say flip it, okay, is if this is a 3' here, yani this is going to be a 5' here. And if this is a 5' here, this is going to be a 3' here. That's what they mean when they tell you flip it. They don't mean start in the opposite direction.
So we just flip the ends.
Just flip the numbers. And I'll show you in the maybe this example is more clear. يعني هنا. This is our template strand. This is the example I gave you guys, right? So if I didn't give you the coding strand مثلا, I only give you the template strand, وقلت لكم ايش mRNA, حتى لو ما حطيت لكم ايش 5' ايش 3', if I tell you template strand, you know the direction it's going in, because this is what we need to copy off of. We know our new mRNA is always 5' to 3' because that's the only way this enzyme works. So then you tell me, and okay, my new mRNA sequence is instead of T and the AA, instead of AU, CC, etc., etc. And this new molecule, بعد ما أكتب, after I match up every single base pair, then instead of saying this runs 3' to 5', I flip the numbers. It's running 5 to 3. That's what they mean when they say flip it. Is that clear?
تمام شكرا.
You're welcome. So RNA polymerase always goes 5' to 3'. Yes, the new RNA molecule is always written like this, 5 to 3. Okay, because it can only copy in the 3' to 5' direction. And
I have a question.
Yeah, of course, Haya.
What if he like asked me about a coding strand? Like he gave me a coding strand and asked for an mRNA instead of giving me a template strand.
Okay, very good question. So remember, what when we say coding strand, what does that mean? It means it looks like our new mRNA, right? But Aya, can you tell me what's the difference between DNA and RNA that we need to keep in mind here?
That RNA has a uracil instead of a thymine.
Perfect. RNA has a uracil. So مثلا, if I had this question, but I didn't give you the template strand, I gave you the coding strand only, إيش حتسوي? It's the exact same, it's its sister. The only difference is what? Anywhere you have a T, you're going to replace it with a U. Make sense?
Like, isn't this what we did? The last question was it with the template strand, the
احنا
difference.
بالlast question, what we did is we took the T وقلنا what matches the T. T صارت A. Because it's a template. Template you're copying off of. Okay, يعني if you have your template, you're using this and you're saying it's like a puzzle piece. What connects to my T? اللي مفروض نقول معاه T حيكون what's connected to it? A, right? Our A what's connected to it?
U.
And then G is C, etc., etc. فلما تقولي لي ال mRNA based off of the template strand, it's not TT again, it's AA. You're seeing the يعني what would match with it. And then you're changing the numbers. بس افرضي إنه I gave you, imagine I didn't give you a template strand, I only gave you the coding strand. إيش الفرق بينهم? The only difference is here instead of T, okay, I kind of covered the T, you have a U. But the order is the same, صح? A هنا لسه A هنا. A after it's the same. C is here, C is still here. You did not change, you're not saying what matches with this because the coding strand looks like the new strand that we're making. فهمتي علي? And maybe this picture will help
Yeah, I get it. Thank you.
clarify it more. Okay, so if they ask you guys coding strand, what is my mRNA? The coding strand looks like your mRNA, so it's the same. You just replace the T with a U. And حتى it's already in the right direction, 5 to 3. If it's the template strand, you're going to match it like a puzzle. So template strand, the T connects with a U, and T connects with an A connects with a U, etc., etc. And then you just change the numbers. Because the numbers are important because they can confuse you guys. مثلا D and E are both correct answers, or the sequence is correct. But if you choose D, it's the wrong answer. Why? Because your mRNA molecule always runs 5 to 3, not 3 to 5. Okay? So RNA polymerase transcribes the DNA in which direction? RNA polymerase only works in 5' to 3'. I'm gonna show you this picture might clarify better. So you see how here your coding strand is running 5 to 3, the one at the top, and your template is running 3 to 5. And when your RNA polymerase is working in this direction, in the 5 to 3 direction, what does that mean? That means the purple molecule here, our new mRNA, is 5 to 3. That's what they're trying to tell you. So it's adding your bases in this direction. That's why we say it works in 5 to 3' direction. Does that make sense? Does anyone have any more questions? I know it's confusing, and I was really confused about this in first year, but I hope this picture specifically helps a lot. Everything else is just what you guys have on your slides. Okay. Last step is termination. So remember, terminate means to what? To end something. So what happens is basically you get to a terminator signal at the end, and the mRNA molecule falls off. Genuinely, that's what happens. Okay, it's like, khalas, I'm done. And so the product that you have is pre-mRNA. Why pre? Because I know we just did a lot of work and we matched the base sequences and whatever, but you have to modify your RNA in order for it to be perfectly ready for your translation process. I'm glad, alhamdulillah, it was helpful. Noor. So basically, what ends up happening when you have your termination sequence, your hydrogen bonds are just going to reform by themselves in the DNA, and it's just going to wrap around itself again. Okay. And so basically what I told you guys, it's just going to keep going until you get to your terminator sequence. Terminator sequence forms something like a hairpin loop. Basically, what you guys need to know about this is that the sequences exactly after each other are complementary to one another. So it kind of forms a sandwich around itself and it just falls off. And it's not, they don't usually ask questions about it, but عشان you guys understand what's happening. So imagine if you started here with your RNA polymerase, you keep adding. Terminator sequence is just going to be 2 complementary sequences in a row, and then it's just going to cause the RNA to fall off. But one thing I want you guys to remember, because they can ask you about it, and promoter region is non-coding. Remember the Tata box, for example, or the cat boxes? Those you don't code. Your terminator sequence are actually, and you're coding them here. And then it forms this hairpin loop, and then from there you modify your RNA further. Okay, so it genuinely just detaches and falls off. All right. Last thing regarding this is you might see numbers thrown around. So remember how we talked about upstream, downstream, where upstream is basically before the gene, downstream is after the gene. Okay. And so if you have anything upstream, you consider it minus. So for example, this region is -1 from the gene, -2 from the gene. You might see it in some textbooks mentioned. I don't remember them using it a lot in the MOL course, but it's in your slides, so I thought I'd mention it. Anything to the right or downstream is plus. So +2, +3, etc. But the first base transcribed in your RNA is just +1 base of that gene region. So basically where you start, they count that as +1. Anything before is minus, anything after is just +2, 3, 4, etc. Again, I don't think it's that important, but I thought I would mention it because it's in your slides. Okay. So this is just a recap, a summary slide of everything that was going on. You have your transcription factors that goes to your promoter region and then helps your RNA polymerase find where to start. So that's your initiation. Okay. And then after initiation is what? Elongation. Your RNA polymerase starts to add molecules. And remember, it adds on the template strand or your antisense strand so that it looks like your coding strand or your sense strand. Okay. And then that keeps going until you get to your termination point. All right. RNA polymerase moves along the template strand. This is, again, since you guys were confused about this point, in the 3' to 5' direction, because it synthesizes RNA in 5' to 3'. Basically, that's just what we said. Remember the picture where I showed you the blue? The new mRNA is 5' to 3'. So it's reading your template strand, which is 3 to 5. Okay. So another thing is RNA polymerase does not proofread its work. It's not like our DNA polymerase enzyme. And transcription ends when it reaches a termination signal. This we already went through. The coding or non-template strand is not used during transcription because it is identical to the RNA sequence. We know this. Again, this is everything that we talked about that we already understand. By convention, the base sequence of the gene found in the coding strand is 5 to 3. Again, ما في شيء يخوف. This is everything we just went through. And then you have specific things called enhancers, which basically show you, it's similar to, it's a type of promoter. So you have your promoter is, for example, here your Tata box, which is short. It's like 30 base pairs. Okay. And then you have an enhancer, which is even further upstream, that basically also helps with regulating transcription of specific genes. Again, this becomes more clinically relevant for you guys when we talk about specific disease processes like cancer and whatnot. Again, these are just, I'm just going through the slides, okay? حقكم. So here you have your gene, before it is your promoter, after it is your terminator, and then you have specific DNA, spacer DNAs or sequences that you don't really code for anything. Yeah, you have a question, Mubarak?
So is the Tata box considered a promoter?
It's within the promoter region. It's the non-coding sequence where you actually start from هرجع لكم. Okay, I'm sorry. I know I'm like going through the slides a lot. I know it's very annoying. I just want to show you guys the pictures because it helps. Yeah. So these are your different promoter elements. Your Tata box, your cat box, your GC box. So remember, your promoters are your repetitive non-coding sequences. It's like the flags. Remember, like the promoters is like the flags telling the pilot you should start your descent here. And the lights are like the different transcription factors that help it find its way. Okay? So basically, that's what you guys need to know about it. So +1 is the base transcript after the promoter region. Yeah, so +1 is the first thing that you start your transcription with. Okay? Do you guys have any questions? We just have
Yes,
Okay.
can I ask question?
Yeah, of course. Go ahead, Omar.
Yes, the promoter doesn't get transcribed. The terminator gets transcribed, right?
Yeah, it gets
Or
transcribed just because it has to fall off كده.
Oh,
يعني literally كأنها سندويشة. It gets transcribed, but there are two sequences ورا بعض that are complementary to one another. So this is complementary to this, so it kind of binds and literally تطبق. It's like a sandwich and then it falls off. يسموها hairpin loop. يعني فعلا تشبه البنسة حقت الشعر. It looks like a bobby pin. That's why they call it a hairpin loop,
Okay,
because it just
بس
binds to
ليش
each other and then it falls off.
ليش بالأخير it's like a circular shape?
ما أعرف عشان كده يسموها بنسة صراحة. بس أهم شيء إنه this is just what you guys need to know is terminator is just where you stop your transcription.
تمام. Thank you.
Okay? You're welcome. طب there's this question for you guys. This is from the slides. فحلوها. I'm just reading one of the questions I got in the chat. So to answer your question, Bilal, yeah, the terminator hairpin is basically what causes the whole thing to fall off and stop transcription. Okay. So Jenna, for the question you asked me on chat, شوفي, if they mention template or if they mention coding, you guys should be able to understand what that means. So I hope the way I explained it to you makes more sense for you guys and what it means when Dr. Ahmed says flip it, or what it means when it says template strand or coding strand. فخلينا نحل هذا مع بعض, and then that'll help us understand if we're going wrong and where. طيب. Now, let's answer the question together. What does a template strand mean? Or what is a template in general in the English language? It's something you're using to copy, to go off of, right? So we know حتى لو ما كتبوا لنا that our template is running 3 to 5. So T, A, oh, that's a really bad A, I'm sorry. G, C, 5 prime. I know they're not putting the prime stuff. I'm just doing this as عشان أساعدكم. So our template is what we're using to copy. So remember, if it's our template, we're gonna find what matches the template so that it looks like our coding strand, because this is our template strand. So what matches T? Guys, عادي إنه say in the chat or unmute. The more you participate, the better it is for you guys. So in RNA, what matches our T? Yes, okay, I don't know why I wrote you upside down, but you guys are correct. How do I do eraser? Okay. Perfect. Okay, so T matches with U. Good. Our A matches with what? our A matches with our, remember guys, لا دقيقة I was right. ليش سمعت كلامكم؟ الله يهديكم معلش. T matches with A matches with U, G matches with C, حلو, C matches with G, right? So now this is our new thing, and we're gonna change the direction. هنكتبها 5 prime to 3 prime. حلو. فإيش حيكون جوابنا؟ Yeah, you have a question about it?
No, I just wanted to note again that the doctor said if he doesn't mention the direction, if it's 5 to 3, it's always gonna be 5 to 3 prime. So it's actually flipped. The TAGC should be, if we write it in 3 prime to 5 prime, is gonna be CGAT. Then we can transcribe it.
Then why would he tell you the template sequence? That's really stupid.
I know, but
Sorry.
he said that
This is going on
he
YouTube.
literally said that. And even
Okay,
the slides.
then والله ما ألومكم إنكم you're confused because that just literally does not make sense. طيب, fine. Let's do it the way Dr. Ahmed al-Jada wants it, although I don't agree with it personally, but I'm here to teach you. I'm not here to agree with his beliefs.
That's
طيب.
weird to be honest, يعني. Because
And this, like, if
template,
that makes sense, why would you tell me it's a template? Like, just tell me this is the sequence. بس okay. طيب. على الكلام اللي انتوا قاعدين تقولوه. إنه Dr. Ahmed is telling you if he writes something, assume it's from 5 prime to 3 prime. حلو. يعني التريك حيكون إيش؟ إحنا we know our template runs how? 3 to 5. So, okay, according to what he is saying, نقلبها. ف 3 prime إنه تكون C G A T. طيب. I'll ask him about this and get back to you guys because يعني, this does not make sense to me. بس if this is the 5 to 3 هذه حتكون 3 to 5. فلما نجي إحنا نقلبها to copy it إيش حتكون؟
It's gonna be GCUA.
Okay, طب you know what I'm gonna do؟ عشان أسهل لكم حياتكم. خليني أشوف I'm gonna discard the annotations. I'm just gonna see what he wrote in the description. Because he said, لا, I was right. يا جماعة. Wait. قلت GCUA is the last thing, right?
Yeah.
Okay, then he said the answer is E, so apparently he wants you guys to do that. I don't understand why, بس مش حالكم. طب شوفوا على هذا الموضوع بما إنه it's really confusing you guys, أنا حسأله أحاول أجيكم على الجامعة بكرة أو شيء أسأله و I'll let you know. Although, again, I don't agree with this personally, but maybe he has a specific way of doing things. So according to him, the answer is E, which is just, you flip it. طب شوفوا خلينا نرجع لهذا السؤال. Okay, بس حاليا خلينا نكمل because I know there's a lot more concepts to go through. I don't want you guys to be confused. Okay, I'm gonna ask him about this and then I'm gonna get back to you. Deal? It's written under the notes. In the notes under the slide, he said the answer is E. Even though, like, to me, that's a little sus. But I think what he's trying to tell you, from what I understood from you guys, is if he writes a sequence and he doesn't tell you what it is, any sequence, like, لو هو قال this is the template sequence, بس ما حط إنه it's 5 prime to 3 prime or 3 prime to 5 prime. He wants you to assume that it's like this. Like the way he wrote it is كذا. T-A G C 3 prime. This is what he's saying. If I give you a sequence but I don't write the numbers, assume it's like this. For what should you do? Because it's a template strand. Template strand isn't 5 to 3, it's the other way. So you have to flip it. You get me? So it's going to be the other way around. So the same thing, but just G C A T 5 prime. So this is basically our coding, or so our template strand. So when we want to copy it, what is it going to be? If we want to turn it into our mRNA, the mRNA, just what matches with C? G, صح? What matches with G? C. What matches with A? U. What matches with T? A. So that's why he's saying the answer is E, not the answer is D. But again, يعني that's do you guys get what he's trying to say here?
Yes.
يعني حتى if he says it's a template strand or it's a coding strand, if he gives you any base sequence, you should always assume that it's written 5 prime to 3 prime. And if he tells you it's template, you need to flip it. Why? Because your template runs 3 prime to 5 prime because they're anti-parallel. Okay? Again, I know it's not like the most straightforward thing, but again, just to repeat it, hopefully hammer it in one last time. If I give you this question, يعني هو أنتوا شفتوا, the question I gave you was actually like, I told you which direction it was running in, this one. So it was easier. Then I told you, this is the template runs كده. بس مثلا what he's trying to say is لو أنا جبت, if I got you guys, sorry, I don't want to speak in Arabic for those who don't understand. If I got you the question, but I didn't put these here. Okay, I just gave you this. You should assume if I didn't write 5 prime to 3 prime in the directions, that you should assume إنه I'm writing it like this, 5 to 3, and then you flip it. And then you solve, and then you flip it again. That's what he's trying to say. I know it's confusing. Again, I'm going to talk to him about this because
I had a question.
I don't know why, like, it's not that hard, guys. Like, I don't know why he's making it complicated. This is hard for me. Yeah, go ahead, ask your question.
I was thinking if, for example, it's given as if it's not specified, is it a template or a coding strand, and we take it as a 5 prime to 3 prime, right? We could just change the bases instead of like flipping it and then making it again. We could just change the bases because we're going to take it as a 5 prime to 3 prime end.
But he should tell you if it's coding or coding or template.
Yeah, but like if it's not mentioned in the question, we're just going to assume it's 5 to 3, right?
If the numbers are not mentioned in the question, you assume it's 5 to 3. Like here, he said template sequence is this. You assume that he's giving you TAGC in 5 prime to 3 prime. But you know that he told you it's a template sequence.
Yeah.
So you know the template runs which way? The opposite, 3 to 5. So then you need to flip it and then you can solve. Then you get E, which is the correct answer. But if I told you this is the coding sequence, what would be the RNA sequence? Then you just assume that this is again 5 to 3 and coding is already 5 to 3. So you don't need to flip anything. Then you just
change the bases.
A, U, yeah, you just change the bases. So it would be the same as this, but instead of T, you have U.
U, yeah.
Yeah, again, but again, if he says it's template, you need to flip it and then solve, which is, I don't know why, but I will talk to him about it and then I'll get back to you guys. I'll make sure to ask Shahad or the PAL team to send it on the back group. But that's basically what you guys were trying to say, you know. If he gives you this, assume it's 5 to 3. If he tells you it's the coding strand, just match this up and just change T with a U. That's it. If he tells you it's the template strand, you need to put it in the correct direction, which is 3 to 5. So you just take this and reverse it. I hope that clears it up a little bit. And again, I'll talk to him and get back to you on that. You're not confused? Okay, amazing, Amar. I feel like I achieved something. Okay. Altayeb, do you guys have any other questions before we get to the last couple parts? I want to give you a break really bad, but I really don't want to keep this going on for like 2 hours and a half, صراحة. So I'm just going to continue and try to repeat as much as possible. Okay. We basically got to a point where we had our DNA, right? We found the promoter region, we initiated, elongated, and terminated our transcription process. Now we have this pre-mRNA. When we say pre means before. Why are we saying it's a pre-mRNA? Because you need to process it. You need to add some things, fix some things, remove some things in order for it to be completely ready. Exactly. So we can consider it an immature RNA. Okay. And so in eukaryotes, which is the cells we're talking about, like multicellular organisms, the primary RNA transcript needs to be further processed. Makes sense. It's called RNA processing. You're processing the RNA. And so everything we just talked about, the transcription, this was happening inside the nucleus. Now we need to be able to take this mRNA, make it stable, and take it into the cytoplasm, because that's where our ribosomes are, right? That's where proteins are actually made, and that's our end goal. So you have different steps. I'll talk to you guys about H and RNA in a second. So our different steps that we have to add a cap to the 5' end. Why? Because this RNA molecule is really unstable. You need to kind of make sure that it's not going to disintegrate at the end. You're not going to lose any base pairs. So you need to stabilize it, and then you can transport it. Okay? So you need to add a cap at the 5' end, which is, remember, the beginning. And then you add a tail to the 3' end. And we'll see what that means in a second. You have RNA editing, and then you splice out something called introns. So this is our DNA, and we transcribe this DNA into our RNA, right? Our pre-mRNA. So basically, this was within our promoter. For those that were asking, see, the promoter is not transcribed through. It's a non-coding region. Then that turns into our pre-mRNA, but we need to add a cap in the beginning at our 5' end, and we need to add a tail at the 3' end. All right, and then what we're going to do is we're going to just kind of shuffle it around, do some editing here and there, and then we get our polypeptide. Okay, and then after translation. And this is, again, a lot of images telling you the same thing. You have your pre-mRNA, which is what we just made. We're going to add a cap, and then we're going to add a poly A tail. So poly, basically here, poly A tail. Poly means many. Anytime you see any word with poly, it just means a lot. So you're adding a lot of adenine. So it's literally screaming, that's what you're doing here to make sure it doesn't unravel. Cap is methylguanosine cap in the beginning, just to make sure it doesn't unravel here as well. And so all of these, basically all these mRNAs that you're fixing, you're editing before you get to your very, very final product is called HNRNA, or heteronuclear RNA. All right. And so it's just basically not the final product. This is all of the work in progress in order to get your mRNA that you can actually make proteins out of. So this is, again, same thing with just nicer image. If you guys can imagine your shoelaces, you have something at the end called an aglet. And that basically prevents your shoelace from unraveling or just getting ruined over time. And so that's basically the same thing that we're doing to our mRNA. We put our cap, here, our 7-methylguanosine cap at the 5' end, and our poly A tail at the 3' end. And so these are basically like the ends of our shoelaces to make sure that this mRNA doesn't unravel. Okay, but they like to ask, I know, like 5' or 3', مرة بتعبكم الموضوع, I know it's annoying, but you need to remember that your 7-methylguanosine is 5', and your poly A tail is 3'. Okay, so again, this is exact same thing we talked about. It's just in your slides. So you have a 7-methylguanosine cap that you add to the very beginning, to your 5' end. And why do we need this cap? There's basically two main functions for it. First thing is what we just talked about, right? We don't want our RNA to degrade because we just worked so hard making it. And that's one reason. So it helps to protect our mRNA. And the second reason is that it serves as a ribosome binding site. Okay, because you're going to need that when you come to the translation part. So that's our cap, our little hat at the top of the molecule. And then you have your poly A tail at the end of the molecule. And that's attached to the 3' end. And this also protects the molecule against rapid degradation. And it also helps to transport it into the cytoplasm. Okay, so that's basically the two functions of that. Okay, so That's basically step number 1. We made our mRNA, right? Now, step number 2, we made sure it's protected. Now, step number 3, we need to edit it. The same way, like when you would take a video or a picture, you would edit it, fix it up a bit. This is the same thing with our mRNA. You have different regions of your mRNA called exons and introns. And you guys might have heard of this already in high school. So your exons are the parts that actually code for protein. Your introns don't. So basically at this point, remember, we have our cap, we have our tail, and now we have specific regions called exons and introns. Our introns basically get cut out because they're non-coding regions. And because they get cut out and they're not created into proteins, for a very long time, scientists thought that they really weren't that important. But we're actually finding out now, I actually did research on this in the summer last year in Bart's Cancer Institute. These long non-coding RNAs play a big role in how these proteins get expressed and the organization and arrangement of these exons. And that's important for things like cancer progression and development. There's specific expression of these introns in different types of cancer cell lines. That's not something you guys need to look into right now or know about, but I just want you to know that introns don't code for anything, but they're still important. They're not useless. They don't just get thrown in the garbage. They have a lot of various functions that we're still learning about. Okay? And exons code for the protein. So what are you going to do? We're basically going to have this molecule called a spliceosome. So think of like splicing, like slicing. Okay, so it's going to cut up our mRNA molecules and just stick the exons together. Remove the introns and stick the exons. Okay, and so this is what our spliceosome does. Our final mRNA is going to be a sequence of exons only because exons are what we code for. Okay, you guys have any questions so far? Okay, silence is an answer. So okay, Mubarak, go ahead.
The spliceosome is there's an mRNA that functions the same function as it. The small mRNA.
Exactly. So the small nuclear RNAs, remember the table I gave you at the beginning, how there's different functions of different RNAs? The small nuclear RNAs are actually the spliceosomes. That's what they help with. Thank you for bringing that point up. So I'm going to show you guys here. This is again from your slides. We said we have different regions called exons and introns. Introns are not coded for. They're non-coding regions of our mRNA that we need to cut out. Okay? And then our exons start to be bound together. So you can see how here it's all red. There's no light. There's nothing in the lighter color. So this is basically the final mRNA product that's actually going to go to the cytoplasm and form our proteins with the ribosomes. Okay? So complexes of snRNA or small nuclear RNA and protein form the spliceosomes that cut out our introns and then give us this nice final product. Okay? And again, this is just what we talked about. Introns do not code for protein. There is generally no accepted reason for the existence of introns. Again, there's a lot of research being done in this specifically, so they do have a lot of functions. It's just not specifically clear what they do because there's a lot of different things that they're implicated in. Okay, and I'll give you guys an example now. So this is again what intron splicing looks like. Here you have your intron, and then you're gonna cut it out. You see how here it's being cut out? So basically you have this intron that's being left alone. And then you have, these are your exons, and that's your mature mRNA. Okay. So now we come to a process called alternative splicing. And this is actually very important in different processes in your human body. Your next block, inshallah, is going to be CVP. And you're going to find a molecule called troponin. But there's a lot of different types of troponin. It's the same protein at the end of the day, but you have stuff called troponin I, troponin T. You guys might have heard of, for example, CK, like CKMB. And there's basically, you'll find a lot of these proteins with subtypes. And the reason you have these subtypes is you have these different exons, right? Remember, in between them, we had the introns that we just cut out with the spliceosome. But instead of sticking them in the exact same order every single time, they can shuffle them around and put them in different order. And that leads to mixing and matching of these proteins. So you have the same protein at the end of the day. It's coded for by the same amino acids. It's just in a different order. So you get a slightly different variation of the proteins that you're talking about. So this is again what it looks like. If this is our DNA, this is our RNA, the intron get cut out, and you can see how the different organizations of colors basically means you just shuffle the order around. Okay? And that's what we call alternative splicing. You guys took it in histology, perfect. So you can see, for example, IgM versus IgG. Those are different. They're still immunoglobulins, but the order of these is different, so you get a different subtype of immunoglobulin, or troponin I versus troponin T, etc. Okay, this is just, again, what it looks like. So alternative splicing. You can get different orders of the same exons because you're shuffling them around. All right. One of the examples is tropomycin. Okay, so alpha tropomycin gene, or TPM1, is organized into 15 exons on chromosome 15. And the way it's organized, or the way it ends up binding to one another here, is going to form our different subtypes. So you have the mRNA in striated muscle, smooth muscle, fibroblasts in the brain. Okay, it's still coming from the same gene. It's just literally you're shuffling them around for specific purposes and specific body systems. Okay, again, I know this looks like a lot. It's just showing you the exact same concept. So you have TPM1, TPM4, and the only difference between them is just the order that they're organized in. Okay, again, this is yet another picture to show you. This is what I was talking about, your immune globulins. So IgM, IgA, IgG, IgD, they're still immune globulins, IGs, okay, that are important for your immunity. اللي هي بالعربي بإنه المناعة. And the only difference is the order that these exons are organized in. It creates a slightly different structure in the end. Okay. So we're learning about all of this and spliceosomes and whatever, and it's important for us to recognize, especially in MOL. I know it can get very redundant and repetitive and detailed and meticulous, but we need to stop and say, okay, as physicians, inshallah, as future doctors, you guys, why is this relevant to us? Well, these spliceosomes that we're talking about are snRNPs. We know that they're small ribonuclear protein complexes that help in mRNA splicing. Sometimes the body would recognize these normal complexes as pathogenic, or they would recognize them as bad. And so the body would start to degrade them. And when your body attacks itself, that's usually what we call an autoimmune disease. Okay? And one of the examples of an autoimmune disease is systemic lupus erythematosus, or just lupus. And it's a widely known disease, and it presents with different things in all of the body systems. It's a systemic thing. So one of the main features they can get you guys in a question is a young female patient presents with a butterfly rash or a malar rash over here, with a lot of like variety of symptoms. Fever, joint pain, easy bruising, anxiety, depression, all of these different symptoms. You can kind of see that it's almost like they're not connected because they're so random and in such different body systems. And that's because SLE is something that can affect any system in the body. And so the reason we're learning about these spliceosomes is because you can have antibodies developed against them, and that can lead to things like autoimmune diseases such as lupus. Okay. Another example is beta thalassemia. So I'm pretty sure you guys learned about it when you learned about different types of anemias. So you have your normal beta globulin gene, and if you have defects in the splicing process, that can lead to subtypes of thalassemia. So if you have a mutation in the splice site where you're supposed to cut it here, but then you end up not cutting it there, that can lead to different subtypes of your thalassemia. And what happens is that decreases or has completely absent beta chains, which you guys know is important for oxygen transport and whatnot. So this is just, again, another clinical example or another reason why we're learning what we are. There's another interesting disease called progeria. There's actually a guy, I forgot his name, but he has progeria and he made a TED talk. It was really interesting. But they consider it basically the case of Benjamin Button, which was like a guy who aged backwards. And what happens in this disease is you have aberrant mRNA splicing due to activation of a cryptic splice site. So what this means is that splicing process is not as it normally is. It starts to splice in a variety of different locations. And so that leads to a lot of different defects. And then you get these patients with a lot of abnormalities in their body, and they have essentially a very big problem with DNA damage repair, and they have unstable genome. And for that reason, they usually don't live that long. But it's just, again, yet another example of why you guys are learning what you are. And I suggest you guys look up the TED Talk. It's very interesting. And it's nice to learn about these different diseases. Altayeb, okay. Here's a question for you guys. And this is a question that they can get you on the exam. A 22-year-old woman sees her physician for a variety of complaints over the past year. These include fevers that come and go, fatigue, joint pain, stiffness, a butterfly rash on the face, sores in her mouth, easy bruising, and increased feelings of anxiety and depression. A diagnostic blood test is most likely to show antibodies directed against which class of molecules? A, ribonuclear protein complexes, B, DNA polymerases, C, carbohydrates, D, TNA, or E, paroxysmal proteins. Okay, can you guys tell me what this disease is? You don't have to say the whole name, you can just say like the no name. Yes, SLE. So here they're describing a case of lupus. One of the most characteristic features they'll tell you is usually the butterfly rash, and that happens in usually young to middle-aged females. And so remember, what happens in lupus, or one of the processes that happens in lupus, is your body attacks the ribonuclear protein complexes, which is basically our spliceosomes. So you can't splice and make your proteins properly, and that causes your systemic autoimmune disease with a variety of different, clinical features. And that's why they actually call lupus, it's one of the diseases that they call the great imitator, because the symptoms can be so random, it can affect genuinely any body system. So it has a variety of different presentations. So again, this is exactly what we were talking about before. But we need to keep in mind that everything that we were talking about now is for eukaryotic cells, so us and humans. There's also a different process for prokaryotic cells. And it has something to do with something called a Shine-Delgarno sequence. And I'll show you what that means now. So first, let's recap everything we just talked about. We said we started our transcription process, right? We had our elongation, our initiation, elongation, termination. Then we got our pre-mRNA. That pre-mRNA was edited. First, we added a cap, a 7-methylguanosine cap at the 5' end. And then we added the poly-A tail at the 3' end. And then we did RNA editing and alternative splicing. And now we have our final mRNA product that is ready to go and be translated into protein. Okay, so this is just all of the stuff that we talked about. This is, again, just so you guys know from your slides, editing is done by modification, insertion, or deletion of one or more bases. We know that you have uridine in RNA. And what do you guys need to know from here? Let me just go through it. Okay, so basically what they're trying to tell you here is the benefit of all of this editing of RNA is that based on one gene, you can have a lot of diversity, right? Because we're modifying, we're inserting, we're deleting, we have our introns, our exons, our alternative splicing. That basically allows us to create a variety of different proteins based off of just one same gene. So that makes it more efficient in the long run. Okay. And here, so you have insertion, deletion of uridine bases and site-specific deamidation of C to U. So you have different types of deamination. So D means to remove something. Amine refers to like an amine group, and ation is just a process. So it's the process of removing an amine group. So what they're trying to tell you is there are specific RNA editing interactions where you would remove or substitute one type of base for another type of base. So an A to I deamination is one of the common forms that we have in humans. Okay, and it's important when we form secondary structures, basically. So you can see how these different groups would interact with the same type of molecule and then end up having different structures. All right, and this is how RNA editing goes. Again, I know we talked about it generally. You have your hybridization of RNA with gRNA, which is basically a guide RNA. So this is where you can have your insertions and deletions of your specific bases. Okay, and then you have your cleavage of RNA with endonucleases. So you can cut your RNA at different points. And you can have addition of uridine bases as well as removal of them. So again, this is just, in my opinion, general information. If you guys have any specific questions about this, let me know, but it's not the most high yield part of your lecture. Like your RNA ligase, this is just general stuff. Okay. So cytidine deamination is tissue or site specific. So this specific process is actually clinically relevant to you. So you have cytidine, which is your C, right? So you know how you have G, C, A, U, T. So your C deamination is tissue or site specific. What does that mean? That means it happens in specific organs or specific places, but not others. And the reason this in particular is important is because it has it codes for your ApoB100 genes. And this is important for the processing of fats in your body, and it's site specific. So this usually happens in your liver. Okay, so you have your ApoB100 and your ApoB48. And if you have defects in this specific process, it leads to specific types of familial hypertriglyceridemia. Familial meaning it runs in families, hyper meaning a lot, triglyceride meaning, fats, and specifically like specific types of lipids in the blood, and anemia means presence. So if you have defects in these different things, you have very high levels of lipids in the blood. So that's why they're mentioning it here. So again, this is just the further mRNA editing. So you have your apolipoprotein B gene. This is going off of the exact same example that I'm talking about here. Your apolipoprotein B gene. And then this is transcribed in different areas. You have it in the liver and you have it in the intestine. In your liver, it's unedited and then it's translated. So you get your apolipoprotein B100. But in the intestine and in different regions, it is translated. And so this kind of helps mediate the way fats are absorbed and let's say the way fats are absorbed from your intestines. Okay. And the way they're transported in the blood. So that's why they're telling you that you can have the same protein coded in different places, but the way they're altered alters their function slightly. And if you have general genetic problems with these genes or deficiencies in one process or another, that can lead to issues in terms of the levels of lipids in your blood. Okay, so that's the general idea they're trying to tell you here, is based on whether something is edited or unedited in different tissues can have different results and different functions. Okay. Now we talk about therapeutic intervention. I think you guys have heard a lot about CRISPR and DNA editing and how that's being used in medicine modernly. But now there's actually RNA editing. So instead of editing the DNA, you're actually editing the RNA. And this is being used now in central nervous system or neurologic disorders, brain disorders, and spinal cord disorders. and specifically for serotonin receptors. So serotonin receptors in general are important for a lot of disease processes, like for example, depression, anxiety, a lot of mental illness. And so what they're trying to tell you here is that there are novel ways where you can actually target and edit these RNAs in order to have a benefit in the human body. And there's also specific drugs that inhibit RNA editing enzymes, and I'll give you an example in a second. And it's actually being researched a lot now. Even the research I told you about, we would basically take these long non-coding RNAs, which is the introns, and knock them down and see how that actually affected cancer growth and propagation. So it's a very promising field of study. I don't think you'll be asked about it. It's just nice to know if you're interested. Okay, so let's just go through a general recap of everything. We had our DNA. We had our promoter region with our template strand, which we're copying off of, and then our coding strand, which is what the product is going to look like. And so our mRNA is going to be created by our RNA polymerase through initiation by, finding the promoter region, elongation, and then termination. And then this mRNA is going to be modified with our cap, our poly A tail, and then splicing out our introns, doing alternative splicing, whatever post-transcriptional modification. And then it's going to leave the nucleus and be ready to get translated into protein. Okay. So this is another question for you guys that they can get. And just remember, heterogeneous nuclear RNA is just everything before your final mRNA. So from your pre-mRNA until you get to the very final product. So what do you guys think the answer is? The question is, a group of students are discussing the process of modification of heterogeneous nuclear RNA into mature messenger RNA or mRNA. Which of the following steps involves the addition of a methylated guanine nucleotide to the 5' end? Yep, you guys got it. So this is talking about RNA capping. If I told you, if I brought this question in another way, and I asked you, what can be added to the 3' end in order to protect the RNA, what would you choose as the answer? Poly A tail, perfect. Yeah, Okazaki fragments are basically they're more relevant for DNA replication. They're not here in transcription. Transcription initiation has nothing to do with the post-transcriptional modification either. And RNA splicing is a type of modification, but in order to get to this step, you need to stabilize your molecule. So remember that your poly A tail is important to protect, okay? And your 7-methylguanosine cap at the 5' end is important for two things. One, in order to protect the RNA, and two, in order to serve as an initiation site for the start of translation. Okay, do you guys have any questions so far? I know it's been a long lecture. This is the last part. So we're going to be talking about the differences between eukaryotic and prokaryotic transcriptions. Can anyone tell me what's the difference in general between a eukaryote and a prokaryote? Just from basic science.
Prokaryotes, they do not have a nucleus.
Prokaryotes don't have a nucleus, and they usually are single-celled organisms. So the easiest example that comes to mind is bacteria. مثلا. You go ahead, Mubarak. You can.
Prokaryotes have circular DNA. It's not like in humans. It's like a strand.
Definitely, that's another very important point. Prokaryotes have a circular DNA. Eukaryotic cells are basically, our cells, plant cells, what we generally think of as multicellular organisms. That's just to revise the very basic science that we know. And so in transcription, the process is a bit different. You mentioned an excellent point where you said that prokaryotic DNA is circular. So you saw how, for us, in order to get to the point where you have a protein, you needed to start your transcription. You needed to fix your mRNA. You needed to take it outside of the nucleus, and then you needed to form your protein. So it's a much longer process in eukaryotic cells. In prokaryotes, everything is happening at the same time. Your transcription is happening at the same time as your translation. Like genuinely, everything is happening at once. And your DNA is circular, so it's a bit of a different structure here. And membrane bound organelles is another point. So you have a prokaryotic transcription unit is a bit different than your eukaryotic one. Why? Because you don't have your 5' cap that we talked about and your 3' tail. Eukaryotic mRNA that we talked about very extensively today has both. In prokaryotes, the 5' end of the mRNA is like there's no modification. You genuinely just transcribe and then translate immediately. You just immediately make the protein. There's no intermediary step like we have what we learned about today. Okay, so it's all going on at the same time. And the starting point is what we call a Shine-Dogarno sequence. So in prokaryotes, we said that everything is happening at the same time, right? So your ribosomes bind to the sequence I just told you about in the untranslated region. And so what happens is, again, it's going to start to transcribe and translate at the same time. It's all going on in one go. All right? And so the main differences between RNA and or RNA polymerases in prokaryotic and eukaryotic cells is the structure. So you have in prokaryotes something called a sigma that's required to initiate at a promoter. And there's specific subunits that are different. And before we talk about it, why is this important? Like, let's say when you think about bacteria, and I tell you that these bacteria need these proteins to survive, but they're different than the specific kinds of proteins we have in human cells. So how can we benefit from that in a medical sense? You go ahead, Mubarak.
Antibiotics.
Exactly. Specific antibiotics can target prokaryotic protein production and transcription. But because we understand now that these subunits are different, we can say, okay, I'm going to create an antibiotic that only targets prokaryotic cells. Obviously, you don't want an antibiotic to go and kill your own cells. You want to have it specifically for that function. So again, why is this relevant to us? You have prokaryotic transcription inhibitors. So again, this whole lesson that we're talking about transcription. And so you have a drug called rifampin that is used to treat tuberculosis. You guys are going to learn about that later. And so this specifically inhibits the RNA polymerase subunit. So this is our RNA polymerase in prokaryotes. So this is in our bacterial cells. Okay. And you see how once this rifampin binds, it changes the shape of our protein. And because it changes the shape of the protein, it prevents it from being able to do transcription. It's inhibiting transcription. And so thus it affects RNA synthesis and these bacterial cells die. Unfortunately, like bacteria can adapt and modify and change the binding site, so this drug doesn't work anymore. But when it was first discovered, it was highly effective. So it binds to an allosteric site and causes a change in shape. Basically, that's the whole concept. It's changing the conformation of this protein so that it no longer works in the way that it's supposed to for this bacterial cell, and then your bacteria die. Okay, so that's just the general concept of why it's important for us to understand the differences between prokaryotic and eukaryotic transcription. So that's helpful, that's beneficial. But I mean, you guys have probably heard that if you're hiking in the woods or you're going camping or something, don't eat mushrooms. And there's a very good reason for that. One, it's very different for people to tell what types of mushrooms are safe or unsafe. And this seemingly innocent looking mushroom is actually called a death cap mushroom. And it's called that for a good reason. It has a substance called alpha-amantin, which is a toxin released by this mushroom. And it inhibits RNA polymerase 2 in our eukaryotic cells, so our human cells. So basically, instead of the rifampin, which goes and kills the bacteria, this goes and kills us. So that's literally why it's called the death cap mushroom. You're going to not be able to perform your daily bodily functions because all of your proteins are going to stop being made. All of your transcription is going to be inhibited, and you don't have the RNA to make that protein. So it eventually causes cell death and it's toxic to humans. So if you're ever hiking in the woods or going camping, please do not eat mushrooms. Please do not eat wild berries. But yeah, that's basically the stuff you guys should know. So this is basically what we just talked about. I'm gonna read the question for you. A patient is suffering from chills, vomiting, cramping, and was rushed to the emergency department. He had eaten wild mushrooms for dinner that he had picked earlier in the day. His symptoms are due to an inhibition of which of the following enzymes? RNA polymerase 1, 2, 3, telomerase, or DNA primase? B. Yes, remember, it's RNA polymerase 2. This question came from the Lippincott question book as well. So just keep in mind, these little things, these little annoying details, they tend to ask about. This in general, yeah, so that was just the answer that we talked about. Do you guys have any more questions? Like that, we should be towards the end of the lecture. But if there's anything you're confused by, I'm more than happy to stay and explain it for you. Just let me know. I know it's been a long day, but thank you for sticking around. And I hope it was helpful.
Allah يعافيك, it was very fruitful.
Alhamdulillah, Alhamdulillah, Allah يوفقكم. I know MOL is annoying شوي. But inshallah, you guys are gonna get through it with flying colors. Or I know the PAL team is working really hard to provide you with sessions for everything. So hatta if you're confused about anything, you have any questions you want to ask me, feel free to email me. Meaning I'll hopefully get back to you guys within 1 to 2 days. That's thank you. Any last closing remarks from me? This is also the same quote I gave the students in pre-med. Self-respect is the root of all discipline. The sense of dignity grows with the ability to say no to oneself. I know medicine can be really tough, and I want you guys to remember that a lot of medicine, especially in first year, I know it can be scary, I know it can be intimidating, but you really need to try your best to keep yourself on track. And you try to مثلا study week by week, study for TBLs, keep yourself disciplined because there's a lot to do and I know it's tough, but if you're not pushing yourself to do that, no one is going to push you. So make sure you're always keeping yourself in check and genuinely, I say this to everyone as our juniors, if you're struggling with anything, you have any problem, this goes for males and females both, of course. I promise you, if you stop anyone in the library, any one of your seniors, and you tell them, hey, I'm confused about this, can you help me? There's no one that's gonna tell you no, unless they're genuinely very, very busy with something. But we're always ready and happy to help you guys. So please, let us tahaw. If you need help with anything, don't be shy. We know medicine is tough and we're here to support you at the end of the day. And just make sure to also keep yourself in check and know that discipline is infinite, but motivation will not last you forever. So yeah, if you guys need anything, we're always here for you and we wish you all the best. وعقبال ما نشوفكم إن شاء الله أحسن الدكاترة. I think that's my last slide. You're welcome. Rawad, are you still on board? I know it's been a long session, I'm sorry.
Yes, of course.
Okay, I'm gonna send you guys, I know I told you how to send it. I'm just gonna edit one of the questions that was wrong. And if anyone has any questions or need anything, and my email is here and I'm more than happy to help you guys. But I think we're done.